Hello students, today we will learn about the aquaculture. Now in this aquaculture we will be having a syllabus distributed with the prawn culture. In the prawn culture we will be going to study the different species of the cultivable prawns in the marine forms and the freshwater forms. Then we will discuss about the different cultural methods here in the prawn culture. Then the harvesting, preservation and marketing. Then after this, in this topic we will also cover the pearl culture, okay, pearl culture, and then the third part, that is we will study the pisciculture, that is the cultivation of the fishes. So aquaculture, we know that is the aquaculture is the cultivation of the aquatic animals, cultivation of the aquatic animals in a water bodies for the human consumption and the economic purpose, okay, and their economic purpose. That is the controlled process of cultivating the aquatic organisms, especially for the human consumption and as well as for the commercial purpose. Now the aquaculture is similar to the agriculture. Aquaculture is similar to the agriculture, where the farmer cultivates the plants and the livestock on the land. In the same way here, the farmer cultivates the aquatic animals in the water bodies, and that's why this aquaculture is also referred as the fish farming. Okay, it is also referred as the fish farming, and the person who are engaged, they are called as the fish farmers. Okay, they are referred as the fish farmers. In aquaculture, the many different types of aquatic organisms are cultured. For example, catla or rohu fishes, then the prawns, okay, marine forms, and the fresh forms, that is the shrimps and the prawns. Then the oysters, maybe for the consumption, that is edible oysters, and for the commercial purpose, that is the pearl oysters, etc., are cultivated here in the aquaculture. Then the food produced by this aquaculture is called as the aqua food. Now why this is aqua food? Because for the fulfillment of the human need, okay, of a aquatic food, we have to carry out this in the more and more developed way, or in the advanced ways. That is, it has to be done as the green revolution which took for the plants, is it clear, or the agricultural field. Now that is called as the blue revolution in case of this aquaculture. Now here in aquaculture we will be going to study, I said, the first is the prawn culture. Now there is the two terms are coming, that is the one is the shrimp and the prawn. The shrimp and the prawns, which are interchangeably used from many nation to nation. Different nations have the different terminologies for the shrimps and prawns. But according to the TVR Pillai 2005, in his aquatic literature, he referred these freshwater forms, which belongs to the family Pelaminoids, okay, Pelaminoids, as a freshwater forms, okay, as a freshwater forms, whereas the family belonging to the Pinidae, okay, family Pinidae members, that is, they are the shrimps, and mostly they all are the marine forms, okay, they are the marine forms. So here it is clear that the term the prawn is used for the freshwater, okay, freshwater forms belonging to the family Pelaminoidae, whereas the shrimps are the marine forms, they are the marine forms and belonging to the family Pinidae, okay. Now there is a little bit difference between the shrimps and the prawns also, okay. That is, they are the saltwater, shrimps are the saltwater, I told, marine forms, whereas the prawns are the what? They are the freshwater forms. Shrimps are comparatively smaller to the prawns, or the prawns are little bit bigger than this shrimps, is it clear? Then the plate like gills are present in the shrimps, whereas the branching gills are present in the prawns. You will see that there is a curved body in case of the shrimp, is it clear? And there is a straight body structure in case of the prawns. So here we should remember, that is the when we should use the shrimps and the prawns term. Now these are the different species of the shrimps and the prawn, the marine forms, that is the Penaeus indicus. Okay. Penaeus indicus is the one of the most important commercial species, whose young ones are usually fished in the estuaries. Okay, in the estuaries. Estuaries is a place where the rivers go and meet to the sea. There is a mixing of the fresh water with the saline water. Now this Penaeus indicus grows to the size of 230 millimeter. Okay, grows to the size of 230 millimeter, and it is considered as one of the commercial species. Then Penaeus monodon. Okay, Penaeus monodon is the another commercial species which have the good fisheries in the West Bengal and the Orissa, and it grows to the size of 320 millimeters. Okay. Then the Metapenaeus monoceros, it also grows to the 180 millimeter and one of the important commercially. Is it clear? Then the Metapenaeus brevicornis. Okay, Metapenaeus brevicornis. It grows to the size of 125 millimeter and it shows a good fishery in north region of south and east coasts. Juveniles are fished in the estuary. Now these are the what belonging to the marine forms, that is the Penaeus indicus, Penaeus monodon, Metapenaeus monoceros, Metapenaeus brevicornis. Is it clear? So these are the cultivable species. Out of this, the Penaeus monodon is the one which grows to the larger size, that is up to the 320 millimeter, whereas next to this is the Penaeus indicus, which is the Indian form or Indian origin, that is the Penaeus indicus. Is it clear? Now the species of the prawns, that is usually the fresh forms, that is the Palamon styliferus. Palamon styliferus, it is also one of the important fisheries taking place in the Gangetic Delta. Okay, you will find it in the Gangetic Delta, and then the size of this will grow up to the 90 millimeter. Then the second one is the Macrobrachium rude. Okay, Macrobrachium rude is also one of the seasonal prawn. Okay, it is a seasonal prawn, and then you will find it good fishing in the West Bengal and Orissa. West Bengal and Orissa grows up to the maximum size, that is 130 millimeter. Macrobrachium rosenberger. Okay, Macrobrachium rosenberger is the another one which is growing to the size of 320 millimeter and usually a seasonal, that is in the monsoon and the post monsoon months in Kerala. This is the okay. Then the Macrobrachium malcolmsoni is the another one which grows to the 230 millimeters in its maximum size and found in the northeast coast in monsoon months. Okay, in the monsoon months. So the freshwater forms are the Palamon styliferus, okay, Macrobrachium rude, then Macrobrachium rosenberger, then the Macrobrachium malcolmsoni. Then the are the four species which we have studied, which are cultivable. Is it clear? Then the marine prawn culture. Okay, what do you mean by marine prawn culture? That is also called as the shrimp culture. Told the definition: the rearing of the marine forms. Okay, rearing of the marine forms belonging to the Penaeid families is called as shrimp culture, or they are called as marine prawn cultures. These are the crustaceans belonging to the phylum Arthropoda. The two cultivable species here is the Penaeus indicus and Metapenaeus monoceros. Okay, so this is the Penaeus indicus. Okay, a culturable or cultivable species, and another one is the Metapenaeus monoceros. Okay, so these are the shrimps which are cultured in the coastal waters, and it is one of the tastiest and the costliest seafood. Now, in the shrimp culture, okay, in the shrimp culture, we will be going to study the different steps of this culture operations. Is it clear? That is the first one, the seed collections. Okay, seed collection. Then comes, after seed collections, we need to study the different methods of culturing them. Is it clear? So, culture methods, once we have cultured and reach it to the marketable size, we need to remove them from those tanks, that is the harvesting we call. Then once we have taken out from the harvesting tank, we have to preserve them. There are different methods of preservation till it reaches to the marketing. So, the last step here is the making these harvested shrimps to the reach to the market, that is called as marketing. Now, the first one we will discuss about the seed collection. What are these seeds? The youngest ones of the shrimps can be said are called as seeds. The young ones or the youngest ones of the shrimps are called as the seeds. They include Nauplius, okay, they include Nauplius, Protozoa, Zoea, Mysis, post larva and juveniles. See here, as soon as the eggs are hatched, the first stage is the Nauplius. Okay, Nauplius. The Nauplius develops into the Protozoa. The next stage, then the Protozoa into the Zoea, and Zoea into the Mysis, and the Mysis into the post larva, and larva develops into the juveniles, and juveniles into the adults. Okay, into the adults. So these are the young seeds. Are the young ones are what? They are the seeds. They are the seeds. Now, there are different methods of seed collection. There are different methods of seed collection. That is the seed collection from the natural habitat, okay, from the natural habitat, or from the controlled breeding, okay, from the controlled breeding. And the third one is the induced breeding, making them to lay the eggs in the controlled manner or in the induced manner. Now, first one, seed collection from natural habitat. Okay, natural habitat. Here the natural habitat may be what? That may be the estuaries or the backwaters. Estuaries or the backwaters. I told estuaries. Estuaries are the places where the mixing of water takes place. That is, the fresh water goes and mix with the saline water. At that place you can collect, or the sea water, that's the comes to the another end, that is the backwaters we call. In the backwaters, the seeds can be collected. Now, there are two types we can collect, that is, if the post larval seeds are there, then it can be collected by the nylon nets, and if the larvae are there, the cast nets are used to collect the larvae. Is it clear? Post larval, they should be collected by the nylon nets, and if they are the larvae, you should collect with the cast nets. Now, in the paddy fields also the shrimp seeds can be collected. See, in the paddy fields also the shrimps can be collected. And what happens here, whenever there is a high tide, the high tide brings these seeds to the paddy fields. They bring these seeds to the paddy fields and retain in the fields. There is by a special nets kept at the sluice gate. There is a one I will show you in the next slide. That is, the sluice gate is like a band. That is what the constructed by the bamboo, wood, or the cement construction, where the water in and out flow can be controlled by a gate. Okay, controlled by a gate. So whenever there is a high tide, the high tide will take the water into the field, and once if the water comes into the field, the sluice gate can be closed and then the exit can be prevented. Is it clear? But the seeds collected from the natural habitat, that is in the estuaries and the backwaters or in the paddy fields, these seeds collected is not pure. Okay, it is not pure, it is mixed with the weeds. Is it clear? So that is about the seed collection from natural habitat. That is, collection of this seed is from the estuaries or backwaters. But what is important here, the seed collection is based on the availability and season. Okay, it is based on the availability and the seasonalities. There is another one, second one, that is the seed collection from controlled breeding. Okay, seed collection from controlled breeding. Now, what is done in this method? The mature shrimps are collected from the deep sea. Okay, we have in the first one we have collected directly the seeds, that's maybe the Nauplius okay, zoea, mysis, okay, juveniles, etc. But in this one, we are collecting the mature shrimp itself, okay, from the deep sea, okay, from the deep sea, and bringing them and putting into the spawning tanks. There is a tank which is meant for laying the eggs. Spawning tanks are the tanks which are meant for the laying the eggs. Now, the male and female are released in this spawning tank. That is in the 1:1 ratio. One male with one female is released in this spawning tank. In this one, the water is replaced several times in a day and watched for the chasing behavior. Okay, watched for the chasing behavior of these shrimps. There is a different for the different species, that is the time taken for the copulations. Once the copulation takes place, the eggs are released in 3 to 4 minutes. Okay, and when the eggs are released, at the same time the spermatophores are also released. Say once the copulation takes place, the eggs are released in 3 to 4 minutes, and during the same time the spermatophores are also released, and then the fertilization takes place in the spawning tank. The fertilization takes place in the spawning tank. Now the fertilized eggs settle at the bottom. Once the eggs are fertilized, they get settled at the bottom, and the nauplius, I said, the just hatched shrimp eggs are called as the nauplius. It takes place, it is hatched in 9 hours, okay, in 9 hours. This nauplius passes or develops into zoea, zoea into mysis, mysis into post larva, and then into the juveniles. Okay. Now, once they are juveniles are formed, they are transferred to the nursery tanks. See here, I will just in brief seed collection from the controlled breeding. In natural condition, we have collected the seeds directly from the estuaries and the backwater, and the availability was seasonal. Okay. Here we are collecting the mature shrimps from the deep sea. Okay. Once we collected these mature shrimps from the deep sea, we are bringing them and putting into the spawning tanks in 1:1 ratio. The male and female are keeping into the 1:1 ratio, observing their chasing behavior for that copulation. Once this copulation takes place, okay, that is the eggs are released. After copulation in 3 to 4 minutes only, the eggs are released. At the same time, when the eggs are released, the spermatophore also released, and the spermatophores are released. So the eggs and the spermatophores fuse together, that is fertilization takes place in the spawning tank. Once this spawning tank fertilization takes place within 9 hours or in the 9 hours, the egg hatches into the nauplius. The egg hatches into the nauplius. Now this nauplius develops into or passes through, okay, zoea, mysis, post larva, and juveniles. Now once the juveniles are formed, these juveniles are now transferred to the nursery tanks. Is it clear? That is the controlled breeding method. Now seed collection from induced breeding. Okay, seed collection from induced breeding. Now this is a method where the female is made to lay the eggs under laboratory conditions. Okay, obtaining seeds in the laboratory from the gravid females under controlled condition. Under controlled condition is called as induced breeding. Now see here, the mature females are captured from the deep sea, as we studied in the previous slide. Okay, they have already undergone copulation now. They have already undergone copulation and have stored of sperms in the genital organ called as thalicum. Okay, matured females are captured from the deep sea. They have already undergone the copulation and have stored the spermatophores in their genital organ called as thalicum. In the genital organ called as thalicum. Now these shrimps, mother shrimps, are kept in the spawning tank. Okay, they are kept in the spawning tank. Now what is done here? They have the eye stalks. Okay, that is, see here, that is the shrimps have the 2 eye stalks. At the base of the eye stalk, there is a presence of eye stalk ganglion. There is a presence of eye stalk ganglion. Now, this eye stalk ganglion has a presence of releases the hormone called as GIH. Okay, GIH, that is gonad inhibiting hormone. Gonad inhibiting hormones. Is it clear? Now, the gonads inhibiting hormone, what they do? They prevents or slows down the release of the eggs. So, in this method, the eye stalk, okay, that is in the induced to spawn by the removal of one eye stalk is removed. This eye stalk is removed so that the GIH is withdrawn. Okay, the GIH is removed. Means what? There is no secretion of GIH. When there is no secretion of GIH, the eggs are released in maximum number at the earlier stage. Is it clear? So, see here, that is called as eye stalk ablation. That is called as eye stalk ablation. See here. Now, this is the eye stalk. Okay, these are the two eye stalks. Below the eye stalk, there is called as eye stalk ganglion. This eye stalk ganglion releases the GIH. This GIH is an inhibiting hormone. This GIH is a inhibiting hormone. It inhibits the gonads to release the eggs. Is it clear? Now, what is done? This is removed. Okay, this one of the eye stalk is removed, and the removal of one eye stalk is called as eye stalk ablation. Eye stalk ablation. So, in the seed collection from induced breeding, we are obtaining a gravid female, okay, which has already undergone copulation and stored the spermatophores in her genital organ called as thalicum. Okay, thalicum, and there what we do, we have to leave them in the spawning tank. Before that, one of the eye stalk is removed. One of the eye stalk is removed. That is called as eye stalk ablation. I told at the base of the eye stalk there is a presence of eye stalk ganglion. Okay, eye stalk ganglion. This eye stalk ganglion releases the inhibiting hormone, that is GIH, gonad inhibiting hormone. Removal of this eye stalk is called as eye stalk ablation, and then there is a stop of secretion of GIH. The stop of secretion of GIH, promotes the release of eggs. Is it clear? That is called as seed collection from the induced breeding. Then once we have released it into the spawned tanks, okay, the spawned eggs are fertilized by the sperms released from the thalicum, I told just now. Now, after the spawning, the adult is removed from the spawning tank. Okay, once the eggs are laid, what we have to do? We have to remove those adults from the spawning tank. Now, the fertilized eggs, I told, once they are fertilized, what comes first? That is the nauplius. Okay, the larva called as nauplius. The nauplius develops into the zoea. Okay, the nauplius develops into the zoea. See here, that is, the eggs are there. Okay, the eggs are there. The eggs are hatched. It develops into the nauplius. The nauplius undergoes development, that is called as zoea, then the zoea into the mysis, then the mysis into the post larva, and post larva into the juvenile, and then the adolescent and the adults. So, these are the phases of the life cycle. Here you are seeing in this slide, that is the inshore phase and offshore phase. Okay. Juvenile is found near the shore, at the sea, near the shore. Is it clear? That is, the juveniles are found at the sea near the shore, whereas these all other forms, that is the nauplius, zoea, mysis, post larva, adolescent and adults, are found in the offshore, somewhere far at the sea, far from the shore. Is it clear? Now, the mysis develops into the post larva, which develops into the juveniles. The nauplius doesn't require any feeding. The nauplius doesn't require feeding. The zoea is fed with phytoplanktons. See here, nauplius is a non-feeding. Is it clear? Whereas zoea is fed with the phytoplanktons. Mysis is fed with the zooplanktons. Mysis is fed with the zooplanktons, and the post larvae are fed with the Artemia, nauplii, and minced clam meat. Minced clam meat. Now, the juveniles are transferred from spawning tank to the nursery ponds. Once the juveniles are formed, they are again transferred from spawning tank to the nursery ponds. In nursery ponds, the juveniles are reared for 1 month. Now they are ready for culture in the farms, stock ponds, etc., tanks, etc. Now, see here, we have studied the 3 methods of seed collection. Okay, 3 methods of seed collections. Now, after seed collections, we have to go for the